everybody. I am Dr. Onur Çalışkaner from Bioengineering Department. I hope you are all fine and in safe at your homes. And I know each of us missed the face-to-face class lectures, but I think this is also a good opportunity for you. And today I'm gonna mention about the reproduction and cell division types in the terms of general biology course for the first year bioengineering students. Okay, let's start with the what reproduction is. As you remember from our first lecture, reproduction is one of the common property of living things in the world. So this is a biological process which organisms give rise to new individual, new offspring. So by the way, the individuals or organisms, living things, have a tendency to continue their existence. So in other words, each individual organism exists as the result of their parent reproduction. I mean, for instance, you are the result of reproduction of your parents, and your parents are the result of their parents' reproduction process. Okay, so briefly, this process ensure for the organisms to survive and to continue their existence in the nature, in the world. Okay, there are two basic types of reproduction. One is the asexual reproduction and the other one is the sexual reproduction. Okay, let's start with the asexual reproduction. So, asexual reproduction includes only one single parent. I mean, only one genetic contributor for its offspring. So, inheritance, genetic inheritance, the genetic information is only inherited from one individual organism, one parental organism. So this process, asexual reproduction, results with the genetically similar, identical offspring, because as I mentioned, these offspring, daughter individuals, new individuals, are getting their genetic information from only one cell. So they are called as the clones also. And this process, asexual reproduction, generally seen in all prokaryotes and some eukaryotes, but generally a single cell eukaryotic organism. And asexual reproduction is processed by the mitosis division, and there are two types of asexual reproduction. Binary fission is a very simple method of asexual reproduction. Here, a single parental cell basically mainly divide into two, generate two daughter cells which carry the identical genetic information, identical genome from its parental cell. As you see here, binary fission is a reproduction method for the bacteria, generally seen in the bacteria. So as you see here, there is a parental bacteria cell, let's say, and it has a genetic content. Then this genetic content, all of the genome is replicated. You know, replication means the doubling of DNA. So DNA content or genomic content is replicated, doubled, and then by the division of cytoplasm, these identical copied DNA molecules are divided, distributed into these newly formed daughter cells. So as you see here, at the end of the process, at the end of the binary fission or this asexual reproduction, these daughter cell or in other words offspring has the same genetic content, has the same genome with its parental cell. the other asexual reproduction type is the budding. So the budding is a little bit different from the binary fission. Here a parental organism or a parental cell forms a bubble-like structure or forms a bud. So this bud actually it's like a miniature organism, or assume that this is a baby organism. Okay, this is a miniature of this parental organism. So it grows on the parental organism, and then when it fully develops, then it's separated and it begins to survive individually. Okay, now in the illustration you see the hydra. So this is an organism living in the sea, in the waters. As you see here, this is a offspring organism, and it begin to grow, budding, begin to grow by the budding. Then this bud of organism grow and grow. Then when it is fully developed, then it is separated and begin to survive by itself. And also, also it's seen in the eukaryotic organism, generally single cell eukaryotic organisms such as Saccharomyces cerevisiae. As you see here now, miniature or daughter yeast cells are budding, and when they are fully developed, they separated. Now here pay attention again. Here the offspring cells carry the same genetic content, replicated and take genetic content from the replicated DNA or replicated genome of the parental cell. And fragmentation, so fragmentation like budding, but there is a again difference. Please remember budding. In the budding, the miniature organism occurs, and after development it generates a offspring. But here in fragmentation, now only there is not a miniature or baby organism. Okay, here only one of the pieces or one of the fragment of this parental organism can have a potential to grow and develop into a whole organism. For example, in starfish, you know starfish, when you cut a fragment from starfish, then this starfish can reproduce or can fix the cut fragment by the reproduction. And also in some worms, you know, when you cut when you cut the whole organism into the pieces, each pieces have a tendency or have a potential to grow, to give rise to an offspring organism. As you see here, each plant, please pay attention, these are not developing miniature organisms. Okay, these are just fragments, just non-specialized fragments of the organism. Then each fragment can grow up and give rise to a whole organism. So this is the fragmentation, and this is the different from the budding. And the other way of asexual reproduction is the parthenogenesis. As you know, and you will see in detail in later slides, in the sexual reproduction part. But you know, I know all of you know this issue. In normal conditions, embryo is generated from fertilized egg, right? Fertilization means combining egg and sperm together. But in parthenogenesis, embryo develops from an unfertilized, underlined, please pay attention, unfertilized cell. And as you remember from our previous lectures, these reproductive cells or egg cells, sperm cells, shortly gametes, have the haploid set of chromosomes. So the embryo or the organism which is developed by the parthenogenesis have the haploid set of chromosome contrast to the fertilized embryo or fertilized zygote. So this mechanism is generally seen in some insects such as bees or ants and so on. And as I said in the beginning of the asexual reproduction, mitosis hold these process. Mitosis hold these asexual reproduction. So let's check what mitosis division is. So this is a type of cell division. So that division, that mechanism produces two genetically identical daughter cells. And it's a multi-step process which includes alignment of chromosomes, and then aligned chromosomes are separated, and these separated chromosomes are migrated into the daughter cell. So these are the general approach of mitosis division. So there are two parts of mitosis. One is the karyokinesis. Here a nuclear cell division occurs. Or nuclear cell division means a division of the nuclear content. You know, in nucleus there is a genome, so that covers the separation or division of the genetic content or nuclear content. And after that, in the second part, cytokinesis occurs. Now here cytoplasm is divided and two daughter cells are formed. So before karyokinesis and or before mitotic division, there is a preparation stage, because you know cells should be ready for the new daughter cells. The dividing cell, or before dividing, these cells should ensure something for these daughter cells, right? So this preparation stage is called as the interphase. Here cell grows and grows in size, and organelles, new organelles are synthesized for the daughter cells, because you know daughter cells will need some of these organelles. So parental cell should supply and synthesize these organelles and also macromolecules for its daughter cells. And the most important things, the key point of the interphase is the DNA replication. So all the things will be distributed to the daughter cells, but the most important things, or the most important distribution, is the DNA, right? Genetic distribution of genetic content. So DNA should be replicated before the mitosis, because DNA should be divided equally, right? It distributed equally to the daughter cells. So here, and please remember the previous lecture, chromosome structure. So you know, DNA is not a naked molecule in the cells. It it forms a complex by the proteins, and it is tightly packed. So these are called as the chromosome. These protein and DNA structures are called as the chromosomes. And DNA is replicated, DNA is doubled or copied. Then this copied DNA is not separated. Again, it is remain attached to these main chromosome. So these chromatids, or these chromatin fibers, which carry the identical, or which carry the, please pay attention, which carry the copied DNA molecule, are called as the sister chromatids. Okay. Now, as you see here, at the end of the interphase, DNA molecule which is carried on the chromatin fiber is doubled. So now the genetic content is doubled here. And after interphase, after this preparation, all the preparation is completed, then karyokinesis occur in subphases. So these are the subphases. So I will not go in detail due to the limitation about the time, but I try to summarize some key points of these subphases of mitotic division or karyokinesis. So here prophase is the first stage of the mitosis. Here, you know, in the cells, DNA or the packed DNA is not fully condensed. But before the cell division, it should be condensed furtherly. It should be compacted more and more to organize easily. So here, chromatids, these not compacted very well, chromatids not compacted very well, begin to condense furtherly to form the chromosome structure. So chromosome structure is a more tightly packed DNA and protein complex. And then here nuclear envelope begin to disappear by the phosphorylation of nuclear lamina, and spindle fibers, these are the microtubules, begin to be formed. After that, in prometaphase section, now chromosomes are fully condensed. Now they are ready to be separated. Okay. Now the cell can easily organize and easily divide these organized and fully condensed structures. And also, you know, kinetochores, these are the protein structures which hold the sister chromatids together, and they appear on the centromeric region, and here nuclear envelope is completely fragmented. And also now after nuclear envelope is fragmented, now they are released because you know, before that the nuclear envelope covers the genetic content, and they are arrested, captured by the nuclear envelope. But by the fragmentation of nuclear envelope, now centromeres are released. Now they are free, and they begin to move to the middle section, middle part of the parental cell. And the metaphase, the most or the key point is seen in the metaphase is the alignment, chromosome alignment. Now here released chromosomes are aligned in the middle of the cell, as you see here, but this alignment occurs, or as you see here, each sister chromatids face through the opposite poles. Okay, they are not located like that. Okay, so if due to this alignment shape, so now they can easily be separated, right? Because each sister chromatids has the identical or copied DNA. If they were located like that, so it would be hard to separate them, right? It would be hard to pull these sister chromatids, identical chromatids, through the opposite poles. Now, as you see here, it's very easy to separate and pull these identical DNA molecules, sister chromatids, to the opposite poles of the cells. And also microtubules are mitotic spindles or microtubules attached to these kinetochores. These are the protein structure, protein complexes found in the centromeric region. Then these microtubules attach these kinetochore structures. And in anaphase, now these microtubules forced by pulling these sister chromatids, and on the other hand cohesin proteins which is responsible for the holding these sister chromatids in their centromeric region, and they are degraded. Then by the pulling, by the forcing by microtubules, they begin to migrate to the opposite poles. And in the telophase, now when chromosomes are arrive at the opposite poles, telophase begin, and here now the fully compacted chromosome structures are not required anymore. Now they can be relaxed here, so condensation occurs in telophase. Then nuclear envelope encircles the chromatins again, covers the genetic material, and spindle microtubules disappear because it is not required in this section. Then after telophase, cytokinesis start. So now genetic content is divided, but daughter cells will need the cytoplasmic content as well, right? Because there are some macromolecules, organelles, which is required for the survival of these daughter cells. So after the karyokinesis, cytoplasm divided equally, and now each daughter cells have the again identical, nearly identical cytoplasmic content as well as the nuclear content. In animals and in plants, the shape of the formation of cytokinesis differ, but I will not go in detail for these mechanisms. So this is just a brief summary about the all mitosis sections. And sexual reproduction. Now, contrast to asexual reproduction, here offspring is generated from two genetic contributor, okay? I mean two parents, two different organisms, individual organism, okay? So if there are two parents, so it means the offspring will not be the identical, genetically identical to the one of the parents, because some of the genes, some of the genetic content will come from the one parent, and some of the genetic content, half of the genetic content, will come from the other parent. So the offspring will have a genetic content, a mixed genetic content, okay? Not identical, same or not completely identical to one parent or one individual organism. So here each parent produce gametes, some reproductive cells, I will mention, and these gametes contributes these normal or formation of normal body or normal DNA of the organism. So in males, these reproductive cells or gamete cells called as sperm, and in females they are called as oocyte or egg cell. And there is again a key point, a very important point here. The number, while the formation of gamete cells or reproductive cells, the number of chromosome sets are reduced by a different cell division types called as meiosis occur. Okay, so the meiosis ensure the same chromosome number and same genetic content across the generations, okay, all generations. And these reproductive cells, by the combining each other, by coming together, these coming together called as the fertilization. So fertilization occurs, and then the body of an organism begin to develop. So here these are the gamete cells from male and female individual. And please pay attention, this is this illustration for humans, but you can assume the same mechanism for plant or for other animals as well. So you know, in our body cells we have 46 chromosomes, or in other words we have 23 chromosomes but in pairs. Okay, so how these chromosome numbers remain the same among all individuals, all human beings, or during or across all generations. Okay, so the answer is the chromosome number of these gamete cells. So the gamete cells each gamete cell from each parent has the half of these chromosomes. So as you remember, we call these state as the haploid set, right? So only they carry one copy of chromosome, but after coming together, or shortly after fertilization, each gamete cell contribute equally, as you see here, and they form a diploid, or they form a doubled number of chromosome. I mean, now a fertilized egg carry the double or chromosomes, homolog chromosomes in pairs. Okay, and the meiosis division is a must for the production of these gamete cells. If there were no meiosis while the gamete formation, so it would cause some genetic disorders. And here in meiosis division, you know, please remember the mitosis division. In mitosis division, only sister chromatids are separated, but here at the same time, or on the other hand, homolog chromosomes are separated. Okay, and there are two stages. Meiosis 1, so this stage includes the separation of homolog chromosomes, and the meiosis 2 is like with the mitosis division. So here, like with the mitosis division, sister chromatids are separated. Okay, again, cell need a preparation stage for the meiosis also. So this is the interphase 1, so similar with the mitotic interphase. So again, the cell is preparing for the division, but again, please don't forget, chromosomes are replicated before the meiosis division also. Okay, don't forget it. And as you see here, please remember organism, diploid organism, human. Okay, so it carry two copies of identical chromosomes. Okay, identical DNA. So one came from its father and the other one came from the mother. So this is maternal and this is the paternal homolog chromosome. And while formation of gamete cells, so they are replicated, and each homolog now have the identical DNA. So these homologs now these homologs will be separated in the later stages. So meiosis 1, this is also called as a reductional division. Why? Because the number of chromosome sets are reduced by 1/2. Okay, and there are two main phases. Prophase 1. So here this is the stage of meiosis 1. So here these homolog chromosomes, these chromosome pairs are held together by the synaptotemal complex. So these are the protein complexes, and they are responsible for holding these homolog chromosomes together, attached. Okay, and these structure called as the tetrad or bivalent. And there is a very important event in the prophase 1 section. So it is the crossing over. So crossing over is a reciprocal DNA exchange mechanism. So let's check together. Here, this is the paternal, let's say, paternal chromosome, and this is the maternal chromosome. So a limited fraction or a certain fraction of chromosome is transferred to the another one, and at the same time the same identical locus, please pay attention, the same or identical region, I mean which carry the same genetic content, which carry the same alleles, okay, or identical alleles. So the other parts is going to the another homolog chromosome, chromosome pair, and at the end of the process, at the end of the crossing over, as you see here, there is a shuffle, right? So the chromosomes are shuffled. One part is mixed with the another chromosome, and the another part is mixed with the another homolog chromosome. So this is why. So and also please, if there were no crossing over, so we can be nearly same with our one of our parents. Why? Because at the end of the meiosis, each of gamete only will have the one of the homolog chromosome, right? So assume that there were no crossing over. So this is assume that a sperm cell, okay, and the sperm cell will exactly be same with its parental cell, right? But as you see here, the sperm cell which carry a crossed over chromosome is differ from its parental cell. So this mechanism, crossing over, ensure or provide the genetic recombination or genetic variety. Okay. And metaphase 1, so again the homolog chromosome. Now homolog chromosomes are aligned in the middle of the cell. Now homolog chromosome, so the pairs, the identical DNA molecules, or homolog chromosome which carry the identical DNA, face through the opposite poles. Now they are ready to be separated, okay, they are ready to be migration. And this alignment occurs independently, okay, occurs randomly. So this is independent assortment, and this is the, after crossing over, this is the second mechanism for the genetic diversity, okay. Don't forget, one mechanism which allows genetic diversity is recombination or crossing over. The other mechanism is independent assortment of the homolog chromosomes in metaphase 1 section. And on anaphase 1, now homolog chromosomes, spindles are attached in the metaphase 1, and they are forced to move through the opposite poles, and they are separated and begin to move through the opposite ends of the cell. Now please pay attention. Here homolog chromosomes are separated. As you see here, sister chromatids are still remain attached to each other, okay, don't forget. And telophase and cytokinesis occur. Now as you see here, two daughter cells are formed after telophase and cytokinesis, and as you see here, these daughter cells have the haploid set of chromosomes, right? Because in its parental cell, or the parental cell had the both homolog chromosomes, but these daughter cells have only one set of homolog pairs. So at the end of the telophase 1 and cytokinesis, haploid daughter cells are generated. After that, meiosis 2 has just started, and the meiosis 2 is similar with the mitosis 1. Again, the subphases are same. So this is the equational division. Why? Because now the homolog chromosomes are remained, or the number of homolog chromosomes are remained same, but the sister chromatids will be separated here. So this is the phases same with the mitosis, as I said. So here, pay attention. Here, as you see here, in meiosis 1 or metaphase 1, homolog chromosomes, homolog pairs are aligned. Here, now again homologs are aligned, and anaphase, in anaphase 2, sister chromatids are separated. Okay. So at the end of the process, please remember, at the end of the telophase 1, we have two daughter cells, two haploid daughter cells, and then each of these haploid daughter cells will take a mitosis division, and so two daughter cells from one of these cells and two daughter cells, again, but remember haploid cell, two daughter cells will be generated from the other daughter cell. So at the end of the meiosis, there will be four, take care, four haploid daughter cells at the end of the total process of meiosis division. So the meiosis division or reproductive cells, as I said, are produced by meiosis division. So it takes a special definition for the production of these gamete cells by the meiosis. So in females, the production of egg cells, haploid egg cells, by the meiosis division, called as the oogenesis, and in males, the formation of sperms or the meiosis division, which result with the formation of haploid sperm cells, are called as the spermatogenesis. In general, they are called as the gametogenesis. Okay, so as you see here, at the end of the, so these are the primary oocyte or primary spermatocyte. These are the precursor cells. Okay, you can think like stem cells, but they are not actually stem cells. So they are precursor or main cells, and they are the diploid cells. These precursor cells are diploid cells, and by the meiosis division, as you see here, this is meiosis 1. At the end of the meiosis 1, the number of chromosome sets are reduced by 1/2, and mitosis division or meiosis 2 division occurs. As you see here, each haploid daughter cell gives rise to two daughter cells. So at the end of the process, now there are 4 haploid gametes in females or in males. And these are the some this is the comparison of meiosis and mitosis phases. You can check here. As you see here, in mitosis there is only one section, but in meiosis division it occurs as two parts, meiosis 1 and meiosis 2, and these are the some differences between the stages, and these are the some differences again, but don't forget, in meiosis division crossover occur, homolog chromosomes are separated, and also after that sister chromatids are separated. But in mitosis, homolog chromosomes are not separated. I mean, the number of chromosome sets are not reduced. Okay. Okay, that's all for today. So I hope it would be useful, this presentation would be useful for you, and if you have any question you can contact with me by email or stick messages, and I hope all of you healthy days, and you know in 2 days later there is a feast of Ramadan, so I hope all of you a holy, happy and healthy feast of Ramadan. Okay, take care of yourselves. See you.